Hello, and welcome to Improving Casework Workflow Efficiency Without Compromising Quality, Automated Methods and Data-Driven Solutions, brought to you by Forensic and sponsored by Thermo Fisher Scientific. My name is Michelle Taylor, Editor-in-Chief of Forensic, and I will be your moderator. For today's webinar, you can earn 1 hour of continuing education credit. Following the conclusion of the webinar, you will receive an email with information on how to obtain CE credit documentation. We have a great lineup scheduled to present to you today, but before we begin, I'd like to take just a moment to cover a few logistics. At the end of the presentations, we will hold a question and answer session. To ask a question, click on the Ask a Question tab in the upper right corner of your screen. Please also take note that the right side of the screen features an overview of today's webinar, as well as more information about our speakers. If you have a technical question during today's event, click on the Test Your Connection button on the bottom of the screen. From there, you can access additional webinar support. We also invite you to use the social media widgets beneath the webinar to share with your friends and colleagues. Today, Kaylee Carrillo, a doctoral student from Sam Houston State University, will present data from a recent study that assesses the efficacy of the Applied Biosystems HID Nimbus Presto QNA system in automating the quantification and amplification workflows for forensic casework samples. Kaylee's research interests include optimizing DNA collection, extraction, and enhancement methods for low template samples. Prior to attending Sam Houston, Kaylee received a Bachelor of Science in Biology from the University of North Texas. You will also hear from Laura Riccardi, a Senior Technical Application Specialist within the Forensic Science Application Group at Thermo Fisher Scientific. Laura spent 5 years working in forensic academia and has been a part of the HID team for nearly a decade. In her HID role, Laura leverages her technical expertise, forensic knowledge, and customer focus to significantly support HID automated systems. Prior to joining Thermo Fisher Scientific, Laura earned a PhD from the University of Bologna.
Good morning, afternoon, evening, depending on where you are in the world. My name is Laura Riccardi, and I am a Senior Technical Application Scientist in the Forensic Science Application Group at Thermo Fisher Scientific. Today, I want to talk to you about the performance of our prep filer DNA extraction kits in the GenUp DNA extraction efficiency test. The data I am presenting has been generously provided by Dr. Carsten Ohoff, Managing Director of the Institute of Forensic Molecular Genetics and Director of the GenUp Proficiency Test. The German DNA Profiling Group, originally known as the Stein Commission, was founded in 1980s by the German Society for Legal Medicine. Today, it's a working group of the International Society of Forensic Genetics, and their primary role is to organize proficiency tests for DNA forensic laboratories. The GenUp proficiency tests are among the largest Stein DNA profiling quality control exercises globally. The GenUp DNA Profiling Group partners with the Institute of Forensic Molecular Genetics. This institute receives the extracts along with details on the extraction methods and elution volume used. They perform the quantification experiments and report mean yields and standard deviation to participating laboratories for them to compare their extraction performance. The data I am presenting spans 7 years and 14 proficiency tests, involving 47 laboratories from academic, governmental, and private sectors. Each laboratory received 3 replicate samples containing 10 microliters of diluted blood to extract for a total of 2,016 samples. And when these samples were quantified in duplicates, the total number rose to 4,032 samples. Over the years, both manual, express, and automated prep filer DNA extraction kits were used, though not all kits were used each year or in every test cycles. Overall, the Thermo Fisher Scientific prep filer DNA extraction kits demonstrated superior performance, exhibiting the highest average DNA quantity in 13 out of 14 modules for all sample and inputs tested. Among all the vendors evaluated, the Thermo Fisher chemistry consistently outperformed the alternatives, reaffirming their ability and effectiveness in DNA extraction processes. Looking closely at the subset of 7 proficiency tests focusing on low DNA input, Thermo Fisher chemistries outperformed the other vendors and consistently delivered higher DNA yields. This highlights the robustness and efficiency of Thermo Fisher's DNA extraction solutions, particularly under challenging conditions. This aligns with the next talk, where Kaylee Carrillo will further explore how the HID Nimbus Presto Q&A system complements the PrepFiler automated kits to recover actionable amounts of DNA from challenging samples. Thank you for your attention, and I look forward to any question you may have.
Hello, everyone. My name is Kaylee Carrillo, and I'm a fifth-year PhD student at Sam Houston State University. I'm very excited to be a part of this webinar, and today we will be discussing streamlining casework with the Applied Biosystems HID Nimbus Presto Q&A system. Now imagine this. You walk into lab, it's a great day, you've had your coffee, the birds are singing, and you're feeling like this, very happy. But all of a sudden, you're hit with reality. Deadlines, casework files, an abundance of emails, and now you're feeling stressed and overwhelmed. Well, in this presentation, I hope to give you some strategies to help combat this stress. So in this presentation, I'll be talking about the HID Nimbus Presto Q&A system. I'll be discussing some challenging samples that we ran on automation and comparing it to manual methodologies. I'll be talking about the SHSU field test and closing out with some time-saving capabilities with this instrument. The HID Nimbus Presto Q&A system is a 4-in-1 instrument. It can complete purification, quantification setup, normalization, and amplification setup for up to 96 samples. So this is perfect for a high-throughput lab, which greatly reduces analyst hands-on time. Within the system is the Kingfisher Presto, which is an automated magnetic particle-based purification system that includes magnetic rods that are covered with tip combs in order to conduct multiple wash steps. There is also a turntable system that is working in tandem with the Kingfisher Presto, which can allow for this 96 samples to be processed in 90 minutes. It uses 300 microliter and 1,000 microliter tips. Additionally, there are these core grippers that help with processing your samples throughout the instrument. And like other labs that maybe name their instruments, this really gave us the name for Baymax for our HID Nimbus Presto Q&A system. Within this instrument and software, there are scripts for both PrepFiler and PrepFiler BTA extraction methods. It is important to note that these samples must be lysed manually before being placed on the instrument before purification. The graphic user interface is very interactive and allows for usability for whatever your lab's preferences are. It's very easy to follow, and it can help minimize any risk of setup error. Once you select which protocol you want to proceed with, it will show you a step-by-step guide on how you can set up your instrument, again, minimizing that risk for any setup errors. There are also sample and reagent tracking capabilities that help you know how much is needed for each sample run. For quantification, this system uses the QuantiFiler Trio kit for either the QuantStudio 5 or the 7500 real-time PCR. The only thing to note is that the master mix must be prepared off the instrument. However, the software will tell you how much of each reagent is needed in order to prepare for this run. Additionally, with this system and with this procedure, you need to use the 50 microliter tips, which are color-coded purple for your convenience. Next, for normalization and amplification setup, there are a plethora of PCR kits that you can choose from, such as GlobalFiler, VeriFiler Plus, or even YFiler Plus. And similarly to quantification, the master mix must be prepared off the instrument. So once you decide which PCR kit you want to proceed with, it will give you a similar GUI interface of how it looked for the other protocols. And additionally, this also uses the 50 microliter tips as well. Next, I will be talking about challenging samples that were processed with the HID Nimbus Presto Q&A system and comparing it to a manual workflow. So our samples were collected from STAFS, the Southeast Texas Applied Forensic Science Facility, where we collected from 3 different insults. including surface, burned, and buried remains. And we were able to collect 6 different cadavers for these 3 insults for a total of 18 cadavers. We were then able to sample 5 different skeletal elements per cadaver and complete this in duplicate for a total of 180 bone samples. We were also able to collect hair, nails, and teeth from decomposed cadavers from 6 different cadavers, also completed in duplicate, for a total of 36 samples. Purification was performed on the instrument, while quantification and amplification were completed manually using the Quantifiler Trio and the Verifiler Plus PCR kit. We collected 3 long bones, such as the humerus, femur, and tibia, and 2 different foot bones, including the medial cuneiform, the first metatarsal, or the cuboid, depending on what was available. For our DNA yield per insult, we can see that with our burned samples, it has the largest variability, with some having relatively high yields. On the other hand, our buried samples had a limited spread with lower recovery. And before proceeding to STR analysis, we did have a threshold of 1 picogram per microliter. And out of the 180 extracts, 133 proceeded to downstream processing based on this DNA yield. 57 samples from our surface decomposed, 44 from our burned, and 32 from our buried. Now, this is very representative of real-life scenarios. Bones are notoriously difficult sample matrix, and especially for our buried bones, they tend to be our most difficult insult type here at Sam Houston. But if we look at the comparison of the DNA yield between the HID Nimbus Presto Q&A system and manual extraction, there was no significant difference seen between the surface decomposed or burned remains. However, there was a significant difference between the two for our buried samples, where the HID Nimbus Presto system did do better. This is just showing all of the samples that met that 1 picogram per microliter threshold between automation and manual methods. And as you can see, with the automated method, we were able to do better or even just the same as manual methods. We can see that there is a 17% difference or increase for our surface decomposed and over a 56% increase for our buried samples. So you can see that difference of 133 total samples versus 110 were able to proceed through that pipeline, allowing for additional information to be gathered. And for our autosomal allele recovery, no significant difference was seen between the two methods for our burned or surface decomposed samples. However, for manual extraction, it did do significantly better for our buried samples. But I do want to note that only 18 buried samples met that input threshold with manual versus the 32 samples using the automated method. Next, for challenging samples, we collected hair, which was cut to approximately 3 to 5 millimeters, and once were washed, proceeded with the prep filer protocol. Nails were also cut to 5 millimeters, and once washed, completed that same protocol. For teeth, we collected both premolars and molars. Once they were washed, they were wrapped with a chem wipe and pulverized with a mallet before being powdered with the freezer mill. And this went down the prep filer BTA protocol pipeline. Overall, the highest amount of DNA was actually recovered from our nails. And this can be explained because our sampling came from the nail bed and not just simply the nail clipping. And the nail bed has some adhering soft tissue that then explains the higher DNA yield. But overall, no inhibition was detected, but DNA degradation was observed in our nail and teeth samples, but that is expected, especially since these came from some decomposed cadavers. Now, when we compare the DNA yields between the two methods, hair did significantly better in achieving a higher DNA yield compared to manual methods. And we see that 6 out of 12 samples proceeded through the STR typing pipeline versus 2 for manual. For our nail samples, the automated method also did significantly better than manual extraction, with all of the samples proceeding with STR typing versus 11. And finally, for teeth, there was no significant difference between the two. With the automated method, we had 10 out of 12 samples versus manual, all of the samples were able to meet that threshold. And for our autosomal allele recovery for our challenging samples, over 80% allele recovery was achieved using the automated method. And we did see that samples that were processed with the HID Nimbus Presto Q&A system did produce significantly higher allele recovery for our nails and our hair samples. And on the screen, you can see an electropherogram from one of our nail samples that was processed with our automated method. This was a full profile and something that we always like to see. Next, we tested some touch samples. So we were able to collect swabs from 6 different participants who owned cell phones, keyboards, and water bottles. These samples were not pre-sterilized beforehand, and we had 2 swabs that were moistened with DI water and were used to swab each of the items. 1 swab proceeded with the HID Nimbus Presto Q&A software pipeline, while the other went through manual extraction, but both underwent the PrepFiler protocol. Most DNA was recovered from the keyboards using the automated method, and the lowest average DNA was recovered from our cell phones for both methods. But overall, all of our samples met that threshold for STR analysis. And we did see that touch samples extracted with the HID Nimbus Presto were able to generate extracts with significantly higher DNA yield. And as mentioned, these were not pre-sterilized beforehand or before sampling, so mixtures was something that we did expect. So we saw mixtures for 16 out of 18 samples for our automated method versus 14 out of 18 samples for our manual extraction. But we did see 100% allele recovery from the major contributor or the owner of the item for both methods. We were able to run these samples through StarMix to evaluate the LR value of the major contributor. And we were able to obtain similar or comparable LR values between both methods. Next, I'll be talking about the SHSU field test, where we collected a variety of forensic casework sample types, such as buried, burned, and surface decomposed remains. Some of these samples were the same from our previous study, but we also collected fingernail clippings, chewing gum, hair, a dilution series, and cotton swabs spiked with blood. And this went through the entire workflow from purification, quantification, normalization, and amplification setup that were performed on the HID NP Q&A system and manually in duplicate. For quantification, we used the QuantiFiler Trio kit, and for STR typing, the GlobalFiler IQC kit was used. Our average DNA quantity between the 2 methods was pretty comparable from using the instrument to doing this manually. And here is an overview of our samples, starting with our more high-yielding samples, such as our gum, some of our nails, and our dilution series. But if we take a closer look and zoom in at some of our more challenging or lower quality or quantity samples, such as our bones, we also see comparable yields between the 2 methods. We also wanted to evaluate how well the instrument could make standard curves and compare them to manually made standard curves as well. And we were happy to report that all the metrics resulted as expected, with the R-square value above 0.99 and the slope was within range. We also wanted to look at the replicates for the HID Nimbus Presto Q&A system. Again, looking at these higher quality samples, we did see reproducibility between the 2. Similarly, for our lower yielding samples, of course, there are a little bit of stochastic variations, but overall, we did see reproducible results. For our average autosomal allele recovery between the 2 methods, we did see that this was very consistent between the 2. And next, we wanted to look at the sensitivity series, the mini sensitivity series that we conducted, where we tested 20 nanograms of DNA all the way to 0.625 nanograms of DNA. And we saw that the heterozygote peak heights of the different quantities and replicates were reproducible. Next, in the sensitivity series, we wanted to look at the peak heights versus the peak height ratios, where we were able to observe that they peak height ratios were consistently above 60%. Finally, I do want to talk a little bit more about how much time you can save with using this instrument. So starting with an example of using a bone. Bones are notoriously known for taking forever to process because of the sanding, chipping, washing, and drying processing step. But when comparing the PrepFiler BTA kit manually versus the HID Nimbus Presto Q&A system, if you had 12 samples that you put on the instrument after lysis, because that is the same, if you have 12 samples, manually it would take around 1.75 hours, versus if you put those same 12 on the instrument, it would take approximately 40 minutes. So hands-on time for manual extraction takes around 2.6 hours versus using the instrument is only 30 minutes. So definitely saving much needed time. And also for the field test, we had around 58 samples, and we wanted to also compare the setup time difference between manual and using the instrument. For quantification, manually it took around 50 minutes, but with the instrument, we were almost able to half it with 30 minutes. Now for normalization and amplification, manually this took 1.5 hours, but using the instrument, it only took 35 minutes. So thank you so much for listening and tuning into this presentation. I would just want to give a few shout outs and thank yous to especially Thermo Fisher Scientific for allowing me to be a part of this amazing webinar, the Southeast Texas Applied Forensic Science Facility staffs, because without the donors and their loved ones, this research would not have been possible. The Department of Forensic Science and IFRTI at Sam Houston State University. And if you have any questions, please email me, or you can also find me on LinkedIn. Thank you so much.
Okay, thank you, Laura and Kaylee. Unfortunately, Michelle had to step away, so I'll be moderating today's Q&A session. Just a reminder that there are 3 resources that you can check out by clicking on the resources icon in the upper right of this page. There's a continuing education certificate link, as well as an app note and more info on the HID Nimbus. A friendly reminder that if you have any questions, to click on the Ask a Question button, also in the upper right, and just type in your question, and we will get to as many as we can. Before we begin, though, we're going to have a couple quick few polling questions. So our first polling question, let me pull that up. Our first polling question is, does your lab use automation? And please check all that apply, whether that's DNA purification, quantification setup, STR amplification setup, CE plate setup, all of the above, or maybe not at this time. So go ahead and do that. Click on 1 and or 2 or 3, and I'll give you another 2 or 3 seconds, and then we'll go to the next question. Thank you for doing that. Our next question, I'll ask you right now. Feel free to click on the little X icon in the upper right corner or upper left, and that will close out the poll. Our next polling question is, which area or areas are your greatest need for automation? Check all that apply. Automated differential lysis, DNA purification, quantification setup, normalization, and STR amp setup, or all of the above. So go ahead and click on those, and then go and feel free to hit the X when you're ready. Okay, our last polling question, let me ask this to you. Okay, the last polling question is, would you like to receive more information about the HID Nimbus Presto Q&A system or PrepFiler extraction kits? Check all that apply. Feel free to click one of those options, and I'll give everyone a few more seconds to read through those. Okay, then go ahead and click on the X when you're done, and we will proceed to the Q&A session. So it looks like a few questions have already come in, but by all means, go ahead and send your questions in. Our first question is, how deep were the cadavers buried?
That's a good question.
Unfortunately, at that time, we weren't currently recording those values, but after looking at some of the photos from recovery, we saw that the burials were from 1.5 to around 3 feet.
Okay, thank you for that answer. Our next question is, how long does sample preparation for bones take?
As you saw, that bone preparation can be quite time consuming and it can be lab dependent. So, if you have, let's say, a large window cut, that's going to take a lot longer than maybe a smaller fragment. And maybe depending on the lab, if they're only processing what is the minimum volume for further downstream testing, then that's also going to cut down the time as well. But some labs do a total demineralization method, so that's going to take a lot longer than on our automated methods. But regardless, bones still need to be washed and dried and essentially powdered or put into a fine consistency. And for us, it takes around 2 days from the time that we actually have our sample to finally getting a profile.
Okay, thank you for that answer. Keep those questions coming. Our next question says, why were the buried bones the least successful compared to the other bone insults?
At Sam Houston, our buried bones tend to be our most challenging sample type. We know that with burials, we have, humic acid, which is known to be an inhibitor, and also, of course, these bones are degraded. And we're located in Huntsville, Texas, so we're kind of known as the piney woods area. So those pines have very acidic soil, so that just adds another layer of complications when it comes to downstream processing and adds to what makes them very challenging.
Okay, thank you for that. Does the system have barcode scanning capability?
Yes, the short answer is yes, because the system is equipped with a barcode reader that can be used with barcodes for sample tracking purposes.
All right, thanks. Thank you, Laura. And how do you transfer results from the quant instrument to the HID Nimbus?
This is very simple because once the quantification instrument has completed the run, you can use the export directly from the HID real-time software with the selected samples that you would like to proceed further in the analysis, and that can directly be imported into the HID Nimbus software to be used.
All right, thank you very much. What was your elution volume for the touch samples?
Our elution volume was 50 microliters.
Okay. What is the minimum pipetting volume?
The minimum pipetting volume, technically speaking, is 2 microliter, but a consideration that we need to keep in mind when we are using automation is also the labware where the volume is contained in. And so we have a recommendation that for low volumes, lower than 35 microliter, we recommend to use PCR plates instead of microcentrifuge tubes.
All right. Thank you for that. Can I reduce the cost per sample by altering the dead volumes?
I know this is a good question, but unfortunately not, because with automation, we need to have that volumes. What has been done during the development phase is that we have optimized that volumes, but also the tip counts in a way that the lab cost will be reduced as much as possible.
All right. Thank you for answering that. Keep those questions coming. Looks like another question just came in and said, what should I do if a power outage occurs during a purification run?
So if you run into a power outage or something happens, thankfully there is a log file that will tell you exactly at what step the instrument left off at. And then when you go back into the software, there's actually a few options of what processing step you would like to continue. So it's a run recovery option.
All right. Thank you, Kaylee. Next question is it possible to pause or stop a run on the HID NP Q&A system?
Yeah, so let's say you did happen to forget something, then it's very simple to pause the instrument, make sure you make that everything is good, and you can continue your run.
All right. Thank you for that. Next question looks like it is, are there major differences between bone and teeth prep?
The main difference is their size. With teeth, it's a lot easier to make it into the consistency. You can either use a mallet to pulverize the bone, you can use a mortar and pestle, which you can do that with bones as well. And you could use a chisel as well. We do have enamel in teeth, which does add a little bit of differences, but it still goes through the same processing.
Okay. Thank you for that. Let me just see if there's any more questions that came in. All right, looks like we went through our questions. So I want to thank you, but feel free to ask any more questions and we can answer them via email. We'd like to thank our presenters, Kaylee Carrillo and Laura Riccardi, for sharing their knowledge with us, and also offer a special thanks to our sponsor, Thermo Fisher Scientific, for sponsoring today's event. We'll be sending out the on-demand link for today's webinar about this time tomorrow, so feel free to share that with your colleagues. And once again, thank you and have a wonderful rest of your day.